Okay, next organelle Golgi apparatus, or GIA, and it's made up of the flat membrane called cisterna, and they look like stack of the pancakes on the image. So in here we have 1, 2, this is all stacks, and in here we have this is a lot of this membranous stack like a pancakes together. This is all they together. This is all Golgi in here, and you can see different in here around. This is a little vesicles, what we will talk close to this Golgi. So overall function of the Golgi is to modify different macromolecules and package them into the vesicles. And after this, these vesicles can move these different biomolecules in different area of the cell. So a vesicle is cellular membrane surrounded by the fluid, and inside of this fluid in the vesicles could be different macromolecules, could be protein or lipids, any macromolecules, which is synthesized. So all this is vesicles with different macromolecules inside. So this is called transport vesicles. This is vesicle coming and it's bringing to the Golgi in here. To the Golgi, it's bringing this purple color in here molecules. Then these vesicles will be merged. In here show how they merging in here. So vesicles, again, this is just cellular membrane. This is membrane. So it can merge with the other membrane of the Golgi. And what was inside of the vesicles now will go inside of these long tubes, cisternas of the Golgi. And these purple molecules now in here show they moving through this cisterna. They will move in here through cisterna. And when they moving again on this membrane of the Golgi, what will be enzyme sitting, and they will perform reactions. So something could be attached, something will be removed maybe from these molecules when it's moving. But in the end, new vesicles will be produced, and this is now our purple modified molecules will be detached, separated, like in here, molecules, vesicles inside with molecules, separating, and it can move to the other part of the cell. And in here shows that again, vesicles incoming, they bring in something, then macromolecules will move through this cistern, will be modified. In the end, new vesicles will be produced with inside modified proteins. And these vesicles now, they will have this is on the surface specialized tag, receiving tag, which will indicate where is it need to be transported in the cell. So overview in here show we see part of the nucleus. We see in here rough endoplasmic reticulum, small and smooth endoplasmic reticulum. We see vesicles, for example, protein producing these vesicles detached. They were synthesized, for example, in the rough endoplasmic reticulum. Now these vesicles with proteins moving in the transport vesicles, they will merge. This is a Golgi. To the Golgi, inside these vesicles will move through the Golgi cistern and will be modified. In the end, new vesicles will be produced with these modified molecules, and then these molecules will be moved inside of this, for example, secretory vesicles for the secretion. Now this vesicle will fuse with the cellular membrane in here and release these proteins out of the cell. So this is secretion happening. Secretion. So again, if this is cell pancreas, cells of the pancreas, maybe insulin in here produced and secreted out. So this is overall how these all organelles work together to perform their function and to secrete something. So next, we will talk about vesicles. So again, it's a small, tiny structure inside of the cells, and they are made of the membrane. Yeah, so it's a membrane-bound structure, and inside we will have just fluid with different molecules, for example, or just with fluid. And function of the vesicles to transport these molecules of fluid within of the cell. And vesicles important for different processes, for example, endocytosis or phagocytosis. So when, for example, this is outside of the cell and this is a food, solid food particle or food molecule, some kind of, and cell have pinching like this cellular membrane surround this food molecule, some particle, and this is what become vesicle right now. And this particularly because it's have food inside molecule, it's called phagosomes. Phaga mean eat. Yes, phagosomes, so inside. And then cell will use it to break down and use it for the nutrients. And the other process, pinocytosis. So same way, cellular membrane can just pinch like this, create this invagination fold, and water molecules right now, for example, fluid with dissolved nutrients can go and be separated and become vesicles with this fluid and dissolved molecules and could be used by the cell. And some vesicles can have receptors attached to them. So in this case, receptors is this special 3D shape structure on the surface of the cellular membrane. And when this membrane tries to make a pinch again, invagination to create vesicles, only specialized cells, like in here, star shape, can attach to this receptor. It means now the cell create vesicles which very selective type of the molecule. Only star-shaped molecules could be inside, and it will be delivered to the particular site of the cell when it's needed. Next structure will be lysosome. So lysosome pretty much similar structure with vesicles, only it's usually on the diagrams and different structures. They look very large compared to the vesicles, and they very dark looking, like in here. They're very dark. So this is special. So these lysosomes, large vesicles, kind of, they dark because they contain specialized enzymes inside. They always have these enzymes, and function of the lysosomes to break down different type of the macromolecules to reuse it. So function of these lysosomes to recycle different part of the cell. So for example, if we have damaged molecules, lysosome will absorb it, these damaged molecules. These enzymes will break down and it will be released pieces so cells can synthesize new molecules. Or if we have some damaged cellular organelle, for example, damaged mitochondria here, again, it will be fused together with the lysosomes, and enzymes in the lysosome will break it down. Microorganism. So if cells absorb microorganisms, they will be fused with lysosomes, and enzymes in the lysosomes will break down this microorganism. So when they form, when lysosomes form, so enzymes are produced in the rough endoplasmic reticulum first, then these enzymes will be fused with specialized very acidic vesicles, what show kind of in here in diagram when two vesicles, one with acid inside and the other with specialized enzyme produced in the rough endoplasmic reticulum, they will fuse together, and this is what will become kind of these lysosomes. This acidity is important because enzymes inside of the lysosomes only will function in the acidic environment. Okay, last part what cells have, cellular organelle called cytoskeleton. Like our body have bone skeleton, cells also have cytoskeleton. And it's made up of this long tubular structure, what's shown in here. We have three types of this cytoskeleton proteins, and it's we have actin. It's a smaller size in here. This is the actin. Then we have intermediate filaments. The size of this tubule structure will be medium. And then we have microtubule. Microtubule is a larger diameter tubes. And if we look on this diagram, you will see intermediate filament organized like this inside of the cell. So this is section through the cell, and we see what inside of it. We see this is net between one cellular membrane and the other cellular membrane, net of the microtubulars. Then we will see actin filaments running like a road in here, and we see intermediate microtubulars in here attached to the, in this case, this is to mitochondria they attached. So in order to cellular organelles to move inside of the cell, or different molecules to move, or vesicles they transport different molecules. So in order these vesicles and mitochondria or molecules to move, first they need to be attached to the cytoskeleton. After this, ribosomes or vesicles or mitochondria attached to this cytoskeleton tubules, they can move them into the area where they need it. So function of the cytoskeleton, one will be movements of organelles, macromolecules, whole cell, division. So when cell is divided, was one cell, become two small cells. This is again, we need our cytoskeleton to move everything, separate into side. But cytoskeletons also, how you can see, it's maintain shape and internal organization of the cell, and help cell to carry different reactions, chemical. So all this is different filaments, intermediate filaments, actin, microtubule, all these three types of the cytoskeleton, they have special ability. They can rapidly grow, so it means increase size, or if they don't need it, they can very fast disassemble, separate on the little pieces if cells do not need it. So this is very interesting. So it's very not stable structure, the cytoskeletons, they form these roads and attachment only when cell needed. In here show, for example, different location of different cytoskeletons inside of the cell. Like intermediate filaments will be in here located in the cell. Microtubulars will have different organization, and microfilaments or actin, they in different areas located. They red in here. So on the periphery, microtubulars close to the nucleus, and intermediate filamental all over the cell body in here show. And diameter, you can see this have different size. Smallest actin 7 nanometer tubular, 25 largest. Intermediate filaments 10 nanometer in diameter. This is shown fluorescent microscopy, and this different type of the cytoskeleton again, actin, tubulin, and intermediate. We can have different type, but they again have in here different staining, and we can see this is all green colors. In here show, this is show our location of these different tubes.